Genetic mutations are alterations in the DNA sequence, the very blueprint of life. These changes can arise spontaneously or be induced by external factors. Mutations are fundamental to evolution because they introduce genetic variation, the raw material upon which natural selection acts. Without mutations, there would be no diversity, and evolution as we know it would cease to exist. Induced mutations result from exposure to external agents, known as mutagens. These can include ionizing radiation, such as X-rays and UV light, which can cause thymine dimers. Chemical mutagens, like alkylating agents such as ethyl methane also induce mutations. Biological factors, such as viruses like HIV, can insert into host DNA via retrotransposition. Even oxidative stress from reactive oxygen species can induce mutations, particularly in mitochondria. Mutations provide the genetic variation necessary for natural selection to act. Without genetic variation introduced by mutations, evolution through natural selection cannot occur. Beneficial mutations enhance an organism's survival or reproductive success, thereby increasing its fitness. These advantageous traits spread through the population via natural selection, leading to adaptation to specific environments. Neutral mutations neither benefit nor harm an organism's fitness. They accumulate gradually over generations, contributing to the overall genetic diversity within populations without influencing natural selection. Harmful mutations decrease an organism's survival or reproductive success, reducing its fitness. Natural selection typically removes these disadvantageous mutations from the population over time. The interplay between mutations and natural selection drives evolutionary change. Mutations introduce variation, and natural selection acts on this variation, favoring traits that enhance survival in specific environments. In summary, mutations serve as the raw material for natural selection, continuously introducing genetic variation. This variation allows populations to adapt to changing environments, driving the process of evolution. Genetic drift occurs when random fluctuations in allele frequencies happen in small populations. These chance events can amplify the impact of mutations, causing some alleles to become more or less common purely by luck, rather than selection. Gene flow refers to the transfer of genetic material between populations through migration or interbreeding. This process can spread beneficial mutations across populations, enhancing genetic diversity and potentially introducing adaptive traits to new groups. When genetic drift and gene flow interact with natural selection, they collectively determine the evolutionary path of species. Drift introduces random changes, gene flow distributes genetic variation, and selection filters these elements to drive adaptation over generations. Mutations serve as key drivers of evolutionary change, as demonstrated by several notable examples. Antibiotic resistance in bacteria often develops through mutations in genes such as BLA, which confer resistance to drugs. This can occur via horizontal gene transfer between bacteria. COVID-19 variants emerge due to mutations in the SARS-CoV-2 spike protein, enabling immune evasion and rapid spread. Human lactase persistence, a mutation allowing lactose digestion in adulthood, illustrates positive selection in populations with pastoralist lifestyles. These cases highlight mutations as fundamental to adaptation and evolutionary processes. The future of mutation research promises exciting advancements that will deepen our understanding of evolution and improve human health. Future research will integrate big data and artificial intelligence to analyze complex mutation patterns and predict their effects on organism fitness. Personalized genomics will use individual mutation profiles to develop targeted treatments, such as immunotherapy approaches for cancer, that harness the body's immune system. Understanding how environmental changes, including climate change and pollution, impact mutation rates will be crucial for predicting species responses and managing evolutionary risks. Ethical considerations surrounding germline editing will spark important debates about its potential impact on human evolution and the responsible use of genetic technologies. As mutation research advances, it will continue to illuminate the mechanisms of evolution, offering new tools for conservation, medicine, and our understanding of life's diversity.